Hello Imbibers, a very good evening to all of you. Once again, we are here with another topic from your molecular basis of inheritance. Yes, we have completed few topics from this particular chapter. We have discussed about the discovery of the genetic material. We have discussed about the structure of the DNA, and yes, we have also discussed about the RNA. Today, I am going to actually explain you that how this packaging and of course the replication happens, especially the packaging part is very important. So we're going to discuss that in detail, that how such a long DNA fits in such a very tiny nucleus. Yes, of course, in the prokaryotes as well as the eukaryotes, we're going to discuss that in detail. So before that, let me introduce you to myself. Once again, I'm Anam Khan. I'll be your biology expert for the session 2023 and 2024, and we're going to discuss all the amazing and important topics related to bio, just like we have already completed our first chapter, which is the sexual reproduction in flowering plants, with lots of practice sessions. So without wasting any time, let's start this session. Yes, so children, please, before that, you can join us on the Embibe app and you can register yourself for the upcoming classes, which give you a benefit of before attending the class, you can go and attempt the pre-class activity so that you know what is going to be taught in the class. And after the session gets over, you can always go for the past Embibe activities or the assignments to know whether you have understood the concept or not. Yes, and when we have a practice session, the Embibe app gives you an ability to answer from the Embibe app via laptop, mobile, etc. Yes, which is not available on the YouTube. So let's start our today's session talking about the DNA packaging. As we move forward talking about, children, this is the chapter which is the molecular basis of inheritance, and the topics to be covered in this chapter is the search of the genetic material. Today we're going to discuss about the DNA structure, and we're going to discuss about the packaging, how the DNA is being packed. We're going to next discuss about the RNA world. We're going to discuss about the replication, transcription, genetic code, translation, regulation of gene expression, human genome project, and DNA fingerprinting. I will try as soon as possible so that the chapter gets completed in time. Yes, let's talk about the today's session. As we talk about the today's session, today's concept to be covered is the packaging of a DNA helix, nucleosome, histones, non-histones, chromosome protein, euchromatin and heterochromatin. We're going to discuss that in detail, especially only related to the packaging of the DNA helix. Yes, and children, before we start, let me have a quick recap, because it's been a long time, it's been a rich gap. So let's bridge that gap to have a quick revision session, quick revision slide, quick revision recap. That what we have studied in the previous session, we have studied about the DNA and the RNA were the two types of genetic material found in the living organisms. Yes, the DNA is the genetic material which transfers hereditary characteristics from the parents to the offsprings. Yes, that's why DNA is a hereditary material. Next, what is DNA? DNA is a polymer of nucleotide. Each nucleotide is formed by one molecule of phosphoric acid, a pentose sugar, deoxyribose, and one of the four nitrogenous bases, adenine, guanine, purine, which are the purines, thymine, cytosine, which are pyrimidines. Yes, a DNA molecules is formed of two long anti-parallel chains, which is polynucleotide. Yes, 5 to 3 direction, 5 prime to 3 prime, 3 prime to 5 prime. Yes, and the very first DNA structure was actually determined by the Watson and Crick by double-stranded structure of DNA based on the X-ray crystallography, and of course the Chargaff rule of pairing was basically explained by them only. So this is what we have studied. So of course I'll give you a quick information about this, that if you have missed any session, all the past Embibe classes are already on the Embibe app. You can always watch them whenever you have time. By any chance, if you have missed the session, you couldn't attend the live session, you can always go and watch them. Yes, so let's start our today's session, which is the DNA packaging. So as we talk about the DNA packaging, children, yes, so as I talk about the DNA packaging, you know that a particular DNA is packed inside a very small cell, and how it is packed inside the cell, that is what we are going to discuss, because how do cell packs the long structure of DNA neatly with the nucleus. Can you see this? Yes, this is your whole cell, and this whole cell will have your nucleus, and this kind of a structure, which is your DNA, would be very neatly, very precisely, very cleanly will be wrapped around that. Yes, that will be properly packed inside the nucleus. Yes, so children, this is what we're going to learn in today's session, how this packaging is being done. So as I move forward talking about the packaging, children, yes, So I talk about the packaging as I drag the slider. Let's drag the slider to see the DNA of the cells of a human. So children, as I drag the slider, this is your nucleus. Yes, and I keep on dragging, so this is how the packaging would be done. Can you see this? This is how your packaging could be done. The DNA in one cell is approximately 2 meter long, and the DNA of all the cells of a person put together, it's about 2 times of the diameter of the solar system. Can you imagine how much DNA we have inside our body? Yes. So children, this is just a fact that whole particular cell of a DNA contains 2 meter long of DNA, and if we combine all the cells of a person and put together, it will be a complete round of a 2 times of a solar system. You know how long it would be. Yes, this is just a fact about that this much long DNA is present in the human's body, and this is so clearly, so precisely, so neatly packed inside your cell that it doesn't even feel like that this much long would be the DNA length. Yes, how? That is of course a question. That is what we're going to discuss in today's session, that how cleanly, how neatly this particular DNA is packed inside your cell. As we talk about the packaging, children, first of all, let's talk about what is the length. Yes, we're going to talk about the length in the prokaryotes. We're going to talk about the length in the eukaryotes as well. So as I talk about the length in the eukaryotes and the prokaryotes, talking about the E. coli, which is a prokaryote. Okay, listen carefully. This is something you can always count. You should always know the base pairs, number of base pairs, and you all should always know the distance between the base pairs. To calculate the length of the DNA, 2 things are must. One is the number of base pairs and the other is the distance between the base pairs. Yes, so children, as I talk about the length of a DNA molecule in E. coli, which is a prokaryote, is 4.6 into 10 to the power 6, which is the number of base pairs, multiplied by the distance between the adjacent base pairs, which is 0.34 into 10 to the power minus 9, which is approximately 1.5 mm or 1.56 in 10 to the power 6 nanometer. So yes, you can always calculate the length of the DNA by knowing these 2 things. One is the number of base pairs, the other would be the distance between the 2 base pairs, and you can calculate that. Let's calculate our length of DNA, the human body. Yes, of course, in a man, the length of a DNA in a man. So let's see what we have. We have number of base pairs, which is 6.6 into 10 to the power 9 base pairs. Yes, and I talk about the length of the DNA molecule would be the 6.6 into 10 to the power 9 base pairs multiplied by again 0.34 into 10 to the power minus 9, which is distance between the adjacent base pair. As I multiply them, a particular single man will have 2.2 meter of DNA in a single cell, and in the single human cell a DNA is stretched out. It is almost 6 feet long, contains over 3 billion base pairs, and all of them are accommodated in the nucleus. Just imagine. Yes. So children, of course, this is something which is not easily digested, but this is the truth. This is basically, you can see a single human cell. A single human cell will have a DNA which is stretched out. It is almost 6 feet long. Yes, and contains over 3 billion of base pairs. All of them are accommodated in the nucleus. Imagine. So yes, this is true. This is about the length of the DNA, but now we have to know how exactly this is being packed, that such a long DNA is easily being packed inside the nucleus. So now you know how to calculate the length. Yes, the number of base pairs and the distance between the base pairs, and you can easily calculate the length. As we talk about the packaging, children, what is the most important thing? Yes, talking about the DNA in prokaryotes. As I talk about the DNA in prokaryotes, you know children, prokaryotes are primitive, having a primitive nucleus, but they do have a genetic material which is very different from the eukaryotes. So as we talk about the DNA in the prokaryote, listen carefully. Prokaryotes are without nucleus. Yes, without proper nuclear membrane, you know prokaryotes having a primitive nucleus. Yes, the hereditary material or genophore is a circular molecule of a double-stranded DNA without proteins. Yes, since the prokaryotes are very primitive and do not have a complex body organization, their genetic material is typically circular. Yes, they have a circular molecule of a double-stranded DNA which does not have proteins, which lack proteins. Listen carefully, the prokaryotes have a genetic material which is circular double-stranded DNA molecule without proteins, and DNA is highly folded into a compact mass and it is located in the nucleoid of the cell. Simple. Children, does not have a nucleus, the nucleus lacks nuclear membrane, and then what we call them? We call them as a nucleoid. So this double-stranded circular DNA is packed inside the nucleoid. It is located in the nucleoid. Some positively charged protein hold negatively charged DNA. There are some proteins which are present inside the nucleoid. Please, a very must information you should always know that your DNA is negatively charged. Your DNA is negatively charged, and it will be always attracted towards positively charged molecule. So when the packaging is done inside the nucleoid is having some proteins which are positively charged and your negatively charged. Yes, prokaryotes which have a negatively charged DNA is attracted towards those positively charged, and this is how the packaging is done. So the loops of fold DNA radiates out the proteinaceous scaffold. Yes, as you can even see that this is how basically your DNA is being packed inside the prokaryotes. Yes, they do not have a well organized structure. Yes, they do not have a nuclear membrane which is basically packed. They have a nucleoid which lack nuclear membrane, and sometimes we always say the genetic material lies naked in the cytoplasm. Yes or no, we always say that the genetic material lies naked in the cytoplasm because it does not have a nuclear membrane. Clear? As I move forward, since we have already talked about the prokaryotes which are primitive organisms, but the most complex packaging takes place in the eukaryotes. Let's one by one, I need to click. I'll click the nucleus. Histones, histone octamer, nucleosomes with non-histone protein. Yes, chromatin fiber, and finally the chromosome. So this is how, these are the particular steps. Children, these are the particular steps which basically, this is how the chromatin is basically formed. All of you know the chromatin, which is basically the light and dark side of the DNA. Yes, which is transcriptionally active, some is transcriptionally inactive. Yes, we are not getting into that. We're going to talk about that when we talk about the transcription. Just focus on the packaging. This is how the step-by-step packaging is basically done inside the cell. There is a nucleus, there is a histone proteins or a histone octamer, and this octamer will form a nucleosome. This nucleosome will have a non-histone protein also. Yes, and this is basically how the chromatin fiber will be formed, and this is how the chromosome will be formed. So this is how step by step the formation of the chromosome will be done. This is how the packaging is basically done. Let's talk more about in detail. Yes, you can see that clearly. I'm just waiting. See that clearly. Nucleus, histone, octamer, nucleosome with non-histone protein, chromatin fiber, and this is your chromosomes. Yes, as we move forward talking about the next we have, let's talk about what are histone, what are histone octamer, what is nucleosome, what are non-histone proteins, what are chromatin, what are chromosomes, everything. Whatever you have seen step by step in the previous slide, we're going to explore each and every then in detail. Let's see, let's explore one by one each and every of them. The very first thing comes into the mind, let's explore the histone proteins. Yes, there are 5 histone proteins. Yes, the protein that provides structural support to the chromosome. These are the particular proteins which are providing the structural support. Yes, these proteins are providing structural support to the chromosome. the basic protein with high percentage of basic amino acids, especially arginine, lysine, which are extremely positively charged. Why we need them? This is something which I have already told you, that DNA is negatively charged and we need proteins which are positively charged. So yes, to attract them, to basically, so the DNA could wrap around them. So yes, histone proteins are extremely positively charged and they have amino acids which are very positively charged side chains of amino acid. A group, of course, a polynucleotide chain of the amino acids forms a what? Forms a DNA. Yes, a long chain of amino acid forms a protein. So these side chains of this particular protein or the amino acids are positively charged, and the DNA wraps around the complexes of histone protein to form a compact shape, which plays a very important role in the gene expression. Yes, there are 5 types of histone proteins: H1A, H2B, H2A, H3, H4. Okay, you have to remember this, children. This chapter is absolutely important from your examination point of view, your board examination point of view also, and your NEET examination point of view, if you ever wanted to give your NEET examination. But coming forward, we're talking about the board examination only. So these are the 5 types of histone proteins. One is the H1, H2A, H2B, H3 and H4. So this is about the histone protein which are positively charged, which attracts the DNA, which led to the packaging of the DNA around it because it is negatively charged, attracted towards the positively charged protein. These proteins are made up of amino acids which contains the lysine and arginine, which are having positively charged side chains so that the DNA could easily wrapped around it. Yes, so this is about the histone protein. Let's talk more about, next we have histone octamer. 5 are there, right? Let's talk about histone octamer. So children, see carefully. This is a good 3D structure to tell you. Why octamer? 1, 2, 3, 4, 5, 6, 7, 8. 8 of them are basically forming a group, and this is what you called as an octamer. As we talk about the histone, next we have H2A and H2B, H3 and H4 occurs in pairs to form a histone octamer. Histone octamer binds and wraps around approximately 1.7 tons of DNA, which is approximately 146 base pair. Yes, one histone octamer will wrap around 1.7 ton, not even 2, 1.7 ton, which is approximately 146 base pairs. This octamer only wraps around this. Please remember this octamer of histone protein which will wrap around 1.7 ton. Children, not even 2 tons, 1.7 ton and approximately 146 base pairs. It keeps the genome organized and wrapped neatly within the cell. Yes, this is the reason your DNA is so neatly, so cleanly wrapped around in the cell. 8 histone proteins and DNA comes together to play with a nucleosome. Yes, this is your histone protein. I have wrapped around the nucleus. Together your histone protein and your DNA will make up the nucleosome. Yes, your histone protein which have a DNA wrapped around it, together it is going to make up a nucleosome. Yes, H1 histone or linker histone is a key component of the chromatin. This one. Yes. So ma'am, you said 5 histone protein, but what is the cause? What is the need of H1? So children, H1 is a linker protein. See, H1 histone is a linker histone, is a key component of chromatin. It binds the nucleosomal core particle around the DNA entry and exit side of this, stabilize it. Simple. This H1 protein will help in basically binding of the DNA from the entry to the exit and is basically stabilizing the structure. Yes, The 1.7 ton is complete, but the H1 or the linker protein is the key component of chromatin. It binds to the nucleosomal core particle around the DNA entry and exit site and stabilizes it. So this is what is about the histone octamer. Yes, I hope that is clear. This is about the histone octamer in detail. Yes, shall we go back? Let's go back. Okay. We have discussed about histone octamer. Let's learn about the nucleosome. So, your histone octamer, the DNA wrapped around it. Yes, the DNA wrapped around it. This is clearly called as a nucleosome. So, children, talking about the nucleosome, the structural unit of DNA, packaging eukaryotes, fundamental subunit of chromatin material, consists of a nucleosome core particle along with the linker DNA. So, what is your nucleosome? The octamer plus DNA plus linker DNA. All three of them combiningly formed a stable structure, and that stable structure, what you call it as nucleosome. Each nucleosome core particle consists approximately 146 base pair of DNA wrapped around a histone octamer. A linker DNA approximately have 10 to 60 base pair long. Yes, a linker histone bound to a single nucleosome known as chromatosome. Clear? This is about the nucleosome. A linker DNA has this linker H1, which have certain particular base pairs which are approximately 10 to 60 base pair long. A linker histone brought to a single nucleosome is known as chromatosome. Clear? So, this is about the nucleosome in detail. Yes. Let's go back and discuss about the other. What we have discussed about histone, octamer, nucleosome. Let's discuss about non-histone protein. So, what is a non-histone protein? These green things, these green dots, can you see that? That is your non-histone protein. These are acidic protein in the chromatin which remains after histones are removed. Even if the histones are removed, these are acidic non-histone protein will always remain attached to the DNA. Helps control the process such as DNA replication, RNA synthesis, nuclear transport, action of steroid hormone, transition between interphase and mitosis. It helps making chromosomes further organized and compact. Can you imagine how important this non-histone proteins are? The non-histone protein has a very important task because it helps in DNA replication, it helps in the RNA synthesis, it helps in the steroid hormone action, it helps in the transition phase of interphase into mitosis. Yes, the preparatory phase into the division phase. Yes, it also helps in making the most compact and organized structure. Yes, making it more compact or organized, help in making the chromosome. So, non-histone protein also have their extremely important which remain attached to the DNA and they are absolutely acidic in nature. Yes, clear? Okay, let's move forward. Let's see what we have next. We have chromatin. So, this is your chromatin. Can you clearly see this, children? This is your chromatin. So, this is open and this is closed. Talking about the chromatin, children, a group of nucleosome which is 11 mm thick, which is a chromatin fiber. 11 mm chromatin fiber spirally coiled upon 30 meter thick chromatin fiber known as solenoid structure. These are basically forming a solenoid structure. A solenoid fiber further coils about 300 nanometer fiber which is 6 to 7 nucleosome per ton. Chromatin fiber can unwind for DNA replication and transcription. So, children, chromatin fiber, yes. So, chromatin fiber will have two types, which one, they can be transcriptionally active and they can be open. These are clearly basically forming a compact structure called solenoid. Yes, So this solenoid will have lots of nucleosome inside it. So whenever we need to open the packaging for the DNA replication and transcription, so this chromatin fibers can easily unwind and the DNA replication and transcription can take place. So don't confuse and don't get worried what is solenoid. Solenoid is nothing, is basically 11 mm thick chromatin fiber spirally coiled around 30 mm thick chromatin fiber called a solenoid structure. Yes, both particular chromatin fiber and basically your chromatin fiber thickly wrapped around each other, basically forming a solenoid structure. This is what a solenoid structure would look like. Yes, the chromatin fiber can easily unwind whenever the DNA replication and transcription is needed. Let's go back and talk about the last, which is the chromosome. You keep on talking about the chromatin. This is how your chromosome. So children, the solenoid found after the chromatin further packs into another helix, the super solenoid, which are diameter of 700 nm, they are 700 nanometer. The super solenoid is condensed further to produce a chromosome, is seen in the metaphase. So children, the chromosomes are always be seen when the cell is undergoing the process of division. Yes, these chromatin fibers, these chromatin material condense to form a chromosome like structure. Chromosome packaging. Chromatin packaging requires an additional set of protein collectively called non-histone chromosomal protein. A single chromosomal has stranded DNA helix packed together. A human somatic structure will have DNA packed in 46 chromosomes. Yes, the whole packaging of your genetic material is there in your 46 chromosomes. Yes, 46 chromosome means 23 pair of chromosomes. Yes, so children, everything we have studied right now about the packaging, about the solenoid, about the chromatin fibers, everything is being packed around the typically 46 chromosomes. All this solenoid structure, a typical human will have a 46 chromosomal. The body is constructed as of 46 chromosomes, and all the particular genetic information, all the DNA, all the non-histone protein, solenoid, chromatin fiber is packed around this 46 chromosomes. Yes, next we have, so we are done exploring everything. We have discussed about the histone octamer, nucleosome, non-histone protein, chromatin, chromosomes, and exploring all of the above. So children, what is euchromatin and heterochromatin? This is something I wanted to tell you but was waiting. Chromatin, which is transcriptionally active and transcriptionally inactive. So children, two forms of chromatin are there in the interphase nucleus. Only euchromatin is found in the prokaryotes, whereas eukaryotes have both. Yes, since prokaryotes are simpler organisms, they do not have euchromatin and heterochromatin. It only have euchromatin, sorry, whereas eukaryotes have both. Euchromatin is uncoiled, lightly strained, genetically active region for chromosome. So why? Because this part of the chromatin fiber, this part of the chromatin is transcriptionally active because it is open. It is getting open for the replication and transcription process. But it contains structured gene which replicate during S1 and G1 and S phase of the interphase. Next we have the heterochromatin. Heterochromatin region stains a dark, tightly coiled region as the genes in these regions are metabolically inactive. So children, this particular part which is tightly coiled, which is appearing thick, which is appearing stained, which is not transcriptionally active, those parts are called heterochromatin. So whenever you see a chromatin fibers, whenever you see the chromatin material, you will always observe under microscope that they have light and dark bands. So the light bands are the bands which are transcriptionally active, and the dark bands are the condensed part of the chromatin material which have heterochromatin. So this is the basic difference between euchromatin and heterochromatin. The euchromatin part will always have enzymes for the DNA replication and the transcription processes, while the heterochromatin does not have anything like that. Next we have a question for you. Yes. So children, identify the different levels of packaging. Now drag and drop it to the correct position. Identify the different level of packaging in this. Yes. What is a different level of packaging? Simple. This is the chromosome, this is the histone octamer, yes, and this is your chromatin fiber. Simple, absolutely very simple. Next, 2 hours after lunch, the beta cells of the pancreas produce the insulin hormone. When the chromosome of these beta cells are observed under a microscope, chromatin differentiated into 2 regions, identify the correct region of the chromosome. Simple, so simple. This is the transcriptionally active region. Okay, so children, which brings us to the end of this particular session. What we had a lot of information. We have discussed so many things. We have discussed about histone protein, packaging of the DNA, non-histone protein, euchromatin, heterochromatin, chromatin fibers, nucleosome, packaging in the prokaryotes, packaging in the eukaryotes. So children, this is a lot of information to grasp in one session. I hope you would have understand everything, because we studied that how this long DNA, if all the cells combinedly form a length of the DNA of our 2 rounds of the solar system, is completely easily packed, neatly packed inside your cell. All the information, everything is being contained in 46 chromosome. Yes, histone protein, non-histone protein, everything plays a part in the packaging of the DNA. Yes, so this is all we have studied today. But before you go children, a quick, this is something we're going to study in the next time. Yes, what is this? This is a different diversity. Yes, diversity, everything which is present in your ecosystem. So what do you think? Do all living organisms have the same blueprint? You would think that genetic material of all the organisms are similar. No, we're going to discuss that about in the next session. What is this? This is your Embibe app. This is what I was talking about. You can log in through your phone number and email ID. Then what you have to do, you have to go select your school examination with CBSE, go to the 12th CBSE, select the language, done, and land on this page. Go to the home page button, go to the Embibe live classes and land on this page. This is me right now taking your class. These are the upcoming classes for the next 7 days. Yes. So children, next we're going to discuss about the RNA world. What you have to do, you have to discuss about the, you have to just register and check out the pre-class activities. Okay. Children, you can learn, you can practice, you can achieve everything is present in one single app. Next we have what? Embibe Lens app. You can scan the text, it will give you everything in the 3D, show you everything. It's an amazing visual learning process. We will meet again in the next session talking about the RNA world. Yes, everything, every part of the RNA will be discussed, and then we're going to discuss about the replication in the DNA in detail. Until then, please take care, keep imbibing. Bye bye.
Keep imbibing. We believe in you.